Hello, everyone. My name is Gang Li. I'm a postdoc at Bill Noble's lab here at UW. Today is my great honor to present our recent work, Pseudotime analysis for time series single-cell sequencing and imaging data. As yesterday, we have seen many talks talking about cells are growing asynchronously and also cells differentiate at variable states. Here, I show you a very simple example for a mouse embryonic stem cell differentiation process where cells differentiate to embryonic body cells and all the way to the neuroprogenitor cells. And imagine for a cell, maybe at day 7, you have growth and transit to embryonic body cell, but even another cell at the same time point may grow slightly faster than this cell, and another cell maybe grows slightly slower than this cell. When you combine all the cells at the same time point, look at their cell time, you might see a unimodal distribution. And when you have a time series single-cell sequencing data set, you might have a multimodal pseudotime distribution. And the goal of our project is to infer the pseudotime for such kind of time series sequencing data sets. And the microscopy image coming from the Bonora et al. Genome biology paper. So once you have a pseudotime for a time series sequencing data set, here is a gene by cell matrix. Each cell have a time label. You can use them to identify some time variant genes, the genes that are changing along the pseudotime trajectory, and also you can order your cells based on the pseudotimes. We are not the first one to tackle this problem. People in the field first try the principal components. Usually you'll find your first principal components roughly correlated with the time label you have. Here in this pancreas cells, there are 8 donors. The age of the donor ranges from 1 to 54. You find the first component roughly correlate, but you also find a lot of the models that kind of overlap with each other. And more, there's another method called monocle. If you infer time, you will might heard of this before, but as this example, you will see one of the monocle component is correlated with time, but they put the age 21 on the rightmost, which puts basically the first component doesn't quite reflect the true time label we have of the single cells. So we found that for those unsupervised methods, their first component usually are driven by other global variability. The temporal variability might at the second or the later components might be different from the global variability. Based on that, recently there is a method called Supertime, which adopt ordinary logistic regression, which explicitly use a time label for each cell to train a penalized ordinary logistic regression to select a subset of the genes and to infer the pseudotimes using the time labels. Here is a cartoon illustration where you have a simple score function. Here you have 5 time labels. Supertime will infer 4 threshold for this score function and classify the first group versus the rest, the first two groups versus the rest, so on and so forth. However, when they apply to the same data set, although the performance is much improved compared with PCN monocle, we still see a lot of overlap in this pseudotime model distribution. We motivated by this limited classification accuracy and also the pseudotime inferred by Supertime is rank-based. You see the range of the pseudotime on the x-axis is from -10 to 15. Doesn't quite reflect the true donor ages. So based on that, we propose a new framework called Skeptic single-cell pseudotime classifier, and which basically adopt the multi-class support vector machine model to first infer the class conditional probabilities for each cell, and then infer pseudotime by taking the conditional expectation. And compared with the previous model, we improve our predictive power using not just one score function for all different classes. We will adopt k different score functions. And also our skeptic model would allow for nonlinearity when you consider, say, reader-based function, kernel function to generate nonlinear decision boundaries. The third difference is for the Supertime is showing your training performance on the whole data set where we propose to do nested cross-validation to provide a more robust and prevent the overfitting on the training data set. What we do is like for each data set, we for each fold of the data, for example, the yellow test fold, we will train a model on the purple set and predict the pseudotime on the yellow set and repeat this for the all the data set we have. And we first test our model and compare with Pseudotime and mouse embryonic stem cell differentiation data. On the left is a confusion matrix for our skeptic model. On the right is a confusion matrix for Pseudotime. A higher number on the diagonal indicate better performance of our classification model. And in this comparison, our model achieved a 94% accuracy compared with Pseudotime slightly less than 90%. And the improvement is significant according to a Fisher exact test. And we also want to point out Pseudotime does not have a train test split. It's simply the training performance. So despite the bias towards Pseudotime, we still outperforming them. And you might be curious if our skeptic model are simple learning batch effects or the real temporal structure, because imagine a scenario for each time label, if you have a strong batch effect specific for that time label, you can still train skeptic, achieve a perfect confusion matrix look like this. To confirm that, we perform a leave one timeout analysis. So every time we mask, for example, the T1 time and train a model on T2 and to T5 and predict all the cells from T1. So in a scenario, if the model are simply learning batch effects, we expect the model to predict uniform errors on the off diagonal. So in this case, since we mask the time for each time point, so the diagonal are all 0. And if the model are truly learning some temporal structure, we will expect a successful model to predict time points to be adjacent time point. So if we mask T1, we will expect the model to predict most of the cells to be T2 instead of the rest of the later 3 time points. And when we do this leave one time point analysis, we found indeed our model are predicting the most of the cells close to the diagonal. And this is Pseudotime. And what is a little bit different is that we don't, for the day 11 cells, we don't predict too much on the day 21. This is because the middle 3 time points are embryonic body cells. So they look, for the day 11, they look more similar to day 7 instead of the day 21 neuroprogenitor cells. And on the right-hand side is the Pseudotime visualization instead of confusion matrix. After test out on this data set, we next benchmark our skeptic model on 6 public single-cell RNA-seq data set. 5 of them evaluated in the original Pseudotime paper, which they have shown to outperform PCN molecule. So we directly compare their performance on those 5 data sets. And on all the 6 data sets, our skeptic model achieved uniformly better performance in terms of the classification accuracy, and the improvement is also significant according to Wilcoxon rank test. After showing that the skeptic model can work well on single-cell RNA data, we next test can we generalize this model to single nucleus imaging data. Our collaborator Hunjin from Dr. Fuller's lab here at UW, they help us to collect the same 5 time point differentiation mouse embryonic stem cell differentiation data, but have the single nucleus image. So here I show you some random crops of the image from 5 time points. And specifically, instead of running on 2D image itself, we adopt image processing pipeline, which using the pre-trained residual net to embed all the 2D images to a 1024 dimensional representation. And after we have the 1D vector for each image, we run the skeptic model and compare with Pseudotime. Again, our model can achieve improved classification accuracy, and the improvement is significant according to the Fisher exact test. And more importantly, we find the skeptic model can provide a more fine-grained label compared with the original time point label. For this, we compute the absolute correlation of the pseudotime for each cell with some morphology features. Here I show two of them, nuclear area and nuclear perimeter. We find skeptic pseudotime have a higher absolute correlation. This actually are supported by the literature that during the mouse embryonic stem cell differentiation process, the embryonic stem cell will have large nucleus, and for MBC, they will have smaller nuclei. After showing the skeptic model can work well on the image data, we further test on the sex differences on both single cell attack and single cell RNA sensing data set. So here I show you the male single cell attack sensing results from skeptic model. So they don't have the 5 time point, only 4 for this one. But when we compare it with the female cell performance on the single cell attack, for all the cells of day 7, if we look at the attack pseudotime from skeptic model, we find the female cells have a heavier tail on the left-hand side. This actually is because during the embryonic stem cell differentiation, the female cells show some delayed differentiation pattern, which is well known in the literature. We did the same analysis for the single cell RNA sensing data set by comparing all the RNA pseudotime for the day 7 cells. We observe a similar delay pattern in the RNA pseudotime. And last but not least, we have also tried to extend our framework to the single cell high-C data. For this purpose, we extract the contact decay profile for each single cell high-C data, and then run our skeptic model. We get a pretty decent accuracy. And here are the pseudotime for the 5 time point. You see those models are not very overlapped with each other. Hopefully, I have convinced you our skeptic model can outperform the state-of-art method Pseudotime in both time series single-cell sequencing and imaging data. And we have shown that skeptic can work well for various types of the data set. And it can detect sex-specific differentiation from single-cell attack and sequencing data. In our manuscript, we also show that we can identify some methylation delay using some co-acid data. But with time limit, I will skip this. And our code is available. The manuscript is also available on the bioarchive. With that, I would like to thank my advisor, Bill Noble, and Christine and Xinxian for providing us mouse embryonic samples, and Doc Fuller's lab for generating the images for the analysis, and Jen Philip for the machine learning expertise, and all the Noble Lab members. And thank you for listening. I'm happy to take any questions.
All right, questions for Gong?
Hi, thank you for the super cool method. I was wondering if you could provide any like intuition based on the formulations why your method performs better than Supertime.
Yeah. Yeah, so there are 3 major motivations. The first one, the logistic regression is only one score function with 4 threshold. And for our multi-class SVM model, for each classification, you will have a score function. So that greatly improves the predictive power. And also, logistic regression model is a linear-based model. And with RBF kernel, we can have nonlinear decision boundary, which also outperform the traditional ordinary logistic regression. That's a major motivation.
Thank you.
Yeah, I was just wanted to ask, how would this method compare to like other pseudotime inference methods that try to map out like a continuous time label rather than like these discrete time labels that you use for your model? I was wondering if there's like an advantage over using septic over those kinds of methods.
So just want to clarify, our model have both like a discrete label and also the continuous label. For example, I have shown a plot like this. We have a continuous pseudotime inference by conditional expectation. So we did not compare with other methods like PC and Monaco because in the previous evaluation, Supertime have been shown that outperform other like pseudotime inference methods. So we directly compare with focus our comparison with Supertime in this manuscript.
Nice talk. So I was wondering how your method does if you have very few time points. So if you have, obviously you can't do anything if there's only one.
Yeah.
But if you just have two, then I guess life gets more difficult, and like then how the comparison to monocle and that sort of approach.
Yeah, that's a great question. So for the time series dataset, at least we request at least 3 time points. But when there are only 2 time points, you can still train a classification model. But I'm not sure. We haven't evaluated on such data set, but that's very interesting to look at them.
Feels like in that setting, there should be something that's like in between what you're doing and
Yeah.
what can look at the
Yeah.
trajectory.
Thanks for the suggestion.
All right, let's give Gong another round of applause.